Good afternoon, everybody. It is 2 o'clock Eastern Time, and this is our review session for the exam on Monday. This is weeks 1 and week 2. I see Dr. Brown is with us. I believe we're going to start this time with this week's material, which is the epithelium, and then Dr. Brown will take over and review the material from week to push the record button. Okay. So we are recording. So don't forget, who cannot make it, you can click on the link that we're going to provide you when we receive it, probably later today or tonight, and then you'll be able to listen to the whole session at your leisure. Okay? So let's begin. I'm going to first share my screen. And you should see the title page. Everybody see this? Everybody see the title page?
Yep. Yes.
Thank you. Okay, good. All right. So let's begin. You remember we said initially that there are 4 basic tissue types: epithelial, connective, muscular, and nervous. We are doing epithelial. In the next few weeks, we're going to be covering connective tissue, muscular tissue, and nervous tissue. The characteristic features of epithelial tissue include the fact that they are composed of an aggregation of polyhedral cells. And this is different than the other tissue types, which tend to be more, if they're fixed cells, they tend to be elongated or spindled. So epithelial cells tend to be polyhedral. They're associated with only a small amount of extracellular matrix material. And generally, their functions are lining surface or body cavities and glandular secretion and absorption. A couple of examples we showed. Top image, of course, is skin. And this is an example. Sorry? Do you have a question? Skin is an example of a tissue that provides a covering or lining for the surface of the body. It is a protective surface. Next image down is an example of a tissue that functions as an absorption area, which is the small intestinal mucosa. And finally, on the bottom, we have an image from the pancreas, which is a compound gland that includes both an exocrine gland that produces a secretion, in this case, pancreatic enzymes that are expressed and secreted through a duct system, and an endocrine gland, the islets of Langerhans, that produce hormones which are secreted into the bloodstream. Of course, The most famous of those is insulin. Epithelial cells are characterized by cell shape, by their arrangement in the tissue, and by the property of polarity. So shape, we have a choice of squamous, which are flattened cells, cuboidal cells, which are square in cross sections so that their height is about the same as their width, and finally columnar cells, which are cells that are taller than they are wide. All these cells can be arranged in layers. Histology is a lot of discussion of layers, and epithelial cells can be arranged in a single layer or in multiple layers. And generally, these layers overlie extracellular matrix and connective tissue. Epithelial cells are also characterized by polarity. So each epithelial cell generally has a basal side, an apical side, and in cuboidal and columnar cells, lateral walls as well. One of the characteristic features of epithelial cells is that they sit on a basement membrane. The basal surface of the epithelial cells sits on an extracellular sheet of macromolecules, which we call the basement membrane. This is a semi-permeable filter that we can stain with special stains. In this image, we're showing a PAS stain, and we can visualize it under the light microscope. So all of the red lines that are labeled with small arrows in that image on the upper portion are basement membrane. In the bottom image, we have basement membrane examined at the ultrastructural level. So this is a transmission electron micrograph, 54,000 times magnification. And at this magnification, the basement membrane is resolved into two layers. The more superficial layer is called the basal lamina, and this is characterized by abundant type 4 collagen, as well as some additional proteins, laminins, nitrogen, and perlecan. This membrane is attached to integrins, which are produced and extend from the overlying basal epithelial cells. Just want to point out that Dr. Brown last week, or whoever did your first week's lessons, talked about lamins, which is a protein that occurs in the nuclear membrane. So be aware that this protein in the basement membrane is laminin, okay? Now, the bottom layer of the basement membrane is the reticular lamina, and this is a more diffuse mesh, but the predominant protein is type 3 collagen. So don't get these confused. Reticular lamina is type 3 collagen. Basal lamina is type 4 collagen. This is a chart from Junqueira, and I think you should study this chart because it has lots of important information, much of which may be present on the exam. This talks about characteristic features of epithelial cells, and we've split the epithelial cells into three groups, right? Or sorry, epithelial junctions, which is a characteristic feature of epithelial cells. We split the junctions into three groups: occluding junctions, which is the tight junction, also called zonula occludens; adherent or anchoring junctions, there are three of these: adherens junction or zonula adherens, desmosome or macula adherens, and hemidesmosome. And the last category of intercellular junction is the communicating junctions. We're only talking about one of these, the gap junction. So for each of these, you should look at this table and learn what are the major macromolecules that are involved in each of these junctions. So for the tight junction, the zonula occludens, we should remember that the main protein that is transmembrane linked in this junction are occludens, claudins, and ZO proteins. For the adherens junction, the major protein is E-cadherin. Catenin is also present, but I would particularly remember E-cadherin, which is a very important protein, both in terms of the biology of epithelium, but also in terms of oncology and what we're going to be studying in the future. The next group, the desmosome or the macula adherens, the major protein to remember is the desmoglein. Easy to remember that because they both share the root desmo, both of the words desmosome and desmoglein. Hemidesmosome is associated with integrins. That is the predominant transmembrane protein involved with the hemidesmosome. And for the gap junctions, the communicating junction, you should remember connexin as its constituent protein. Each of these junctions is also associated with cytoskeletal filaments. So remember, the proteins and the junctions are associated with the membrane. The proteins go through the membrane, and on the cytoplasmic side, they're associated with various filaments. The first two junctions, tight junction and adherent, are both associated with actin microfilaments. The desmosome is associated with intermediate filaments, particularly keratins. And the hemidesmosome is also associated with keratin intermediate filaments. The communicating junctions are not actually associated with cytoplasmic filaments associated with the junction. You should know something about the major functions of each of these junctions. The including junctions basically form as a seal. They seal the space between two adjacent epithelial cells. And functionally, what this does is it prevents water and macromolecules and electrolytes from passing between the cells. It forces all those materials to pass through the cells. Transmembrane transport. Transcellular transport. The adherent junctions all function to form a physical link between the adjacent cells or adjacent tissue. So the adherence junction links the cell to its adjacent neighbors, and this junction takes a band-like structure.
I can't run for this shit.
Sorry, Austin, you have a question? Nope. Okay. So the adherence junction forms a band around the cell. In contrast, the desmosome is a spot-like attachment. So it's just a very focal attachment. But both of these junctions serve to form an attachment between the two adjacent cells. Hemidesmosome also forms a structural attachment, but this is a little bit different because of its location. Hemidesmosome attaches the basal cells of the epithelium to the underlying basal lamina. Okay? And communicating junctions have a different function, though. They function to allow communication between adjacent cells. In particular, they allow direct transfer of small.
So our 18, 19, then they come again and see sharp, and see on care, and see on fine.
So in particular, the communicating junctions allow molecules to travel from one cell to another through this transmembrane pore that's created by the gap junction. Major functions. So we've actually. Sorry? Okay. So medical significance is the last row. And the things to pay attention here, we've actually given you a couple of examples of diseases that have to do with issues or problems with some of these junctions. So when you're studying, you should pay attention to those things where we've given you an example of where this junction may have a medical significance. Okay? Yes, Zachary, you got a question?
Yes, hi. Can you just explain the difference between the major function of the adherence junction and the desmosome? Because it seems like they do the same thing, just one is stronger than the other.
Yeah, I wouldn't say they're stronger. They pretty much do the same thing. The one difference is that they're a little bit different. The adherence junction is you'll see the second name for it is zonular adherence. And zonular refers to the fact that it has a belt-like morphology. So that functions to form a tight junction sort of toward the apical end of the cell, just below the tight junction. Okay. In contrast, the desmosome forms spot-like attachments, and they can be scattered generally throughout where the lateral walls are, and often they're deeper in the cell, away from the apical surface. So the function is the same, but the anatomy is different. Okay.
Just to clarify,
Yeah.
I remember in the John Carrot textbook, it said that the order has to be tight junction, zonal adherence, and then desmosome, right?
Yes.
So it's not spread out the way, like meaning it could be spread out anywhere lower than that, you're saying.
Desmosomes can be present in other places, but the what's referred to as the junctional complex, which is the major intercellular junction complex, is just below the apical surface of the cell and actually forms the division between the apical aspect and the basolateral. And in that intercellular junction, it is always a tight junction, adherence junction, desmosome. So if you're given a transmission electron micrograph of that intercellular junction area, and you look from the apical side of the cell down, they should always be in that order: tight junction, adherence junction, desmosome. Okay. Leah, did you have a question?
Yes, I did. Are you able to hear me?
Yep, we can hear you.
I was wondering if you would say that the I don't know how to say the adhesive proteins, like for example, perlecan and nitogen, maybe,
Yeah.
are those equally as important as the transmembrane link proteins?
I would say they're not as important.
Okay.
It's important to be able to recognize them, that recognize the word, but the significant protein in terms of function, we're talking about the basement membrane now, right?
Yes,
I
that,
think
yeah.
the most important thing to remember, because it comes up time and again as you go through this course, and actually if you study pathology also comes up time and again. The most important thing is the type of collagen that's present.
Okay.
So definitely remember that it's type 4 collagen in the basal lamina, which is the superficial part of the basement membrane, and it's type 3 collagen in the deeper reticular lamina.
Okay.
The other ones are important to sort of recognize and know that they're involved. But unless you get into really the molecular biology of the basement membrane, maybe if you're going to be a nephrologist, where the basement membrane is critical, you're going to really get into perlecans and stuff like that. But for now, first pass, those are the really important things. Okay?
Yeah.
All right, so just medical significance, the bottom row, the ones I would point out to you are the adherence junction and the importance of ECadherent, because this has a definite, actually we showed you an example of this. This is very important in terms of the pathogenesis of carcinomas, malignant tumors of epithelial cells. So that's important. And then the other one that is the other two that are important in terms of medical significance at this point are the desmosome and the hemidesmosome, because we gave you examples of how defects in these junctions affect a couple of skin diseases, okay, blistering skin diseases. So those are the ones that I would focus on, okay? Let's see. Okay, so this is a really nice diagram from Junqueira. And actually on the right side, with regard to Zachary's question, this is exactly the image I'm talking about, right? You see this transmission electron micrograph on the right. You'll see that the top of this image is the apical side of the cell. People could trick you by flipping the image around, right? But always look for clues that it's the apex. In this case, microvilli, which we're going to talk about in a little bit, are present at the top of the image, so that's the apical side. And if you go from the apical side down, it's tight junction, adherence junction, desmosome, okay? And this is all drawn in the drawing to the left, okay? Drawing to the left delineates, where these junctions are. And actually, if you'll notice, you see the yellow circles there are desmosomes. And you see that actually there are desmosomes that not only are in this intercellular junction area, but may be scattered in other places of the cell as well. And of course, the hemidesmosome is down here on the basal membrane of the cell, links the epithelial cell to the underlying basement membrane. And gap junctions are scattered usually in the lateral walls because they're connecting to their neighbor cells. Okay. Another view of that famous transmission electron micrograph view of the intercellular junctional complex on the left, and a nice drawing on the right, which clearly documents the junctions and their components. And the other piece of information that it gives you is the associated filaments in the cytoplasm. So you'll notice that both the tight junction and the adherence junction are associated with these lines here, which are actin filaments. And the desmosome is associated with intermediate filaments or keratin filaments. Okay. Desmosome, we've already, I think, hit on a lot of these things, but you'll notice that major protein is desmoglein, okay, which is one of the cadherin family. Desmoglein is a member of the cadherin family, but it is not E-cadherin. Okay, so E-cadherin is associated with the adherence junction. Desmoglein and other cadherin proteins are associated with the desmosome. Okay. You see, I just popped up this histologic picture on the bottom left. This is the picture we showed you in the lab. I think it was in the interactive session about pemphigus vulgaris. So this is an illustration of what kind of a blistering disease we get if we disable our desmosome. So the separation occurs between neighboring epithelial cells, resulting in a cleft that is intraepidermal. It's within the epidermis. Zachary, you raised your hand.
Yes, hi. I just want to know, is it vital that we know the names of all the clinical diseases present, or will we be given a description of what the disease is and work from there, if that makes sense?
Yeah, we're always going to, the name of a disease that we've used for an illustration may appear in a question, but generally the question, particularly at this point in exam 1, the question will be worded so that you don't really have to know anything additional about the disease. You should know whatever it was we were illustrating. Like, for instance, if we give you a question about pemphigus vulgaris, you should know that this involves intracellular junctions, and you should know which intracellular junction is involved. And so you should also know why it gives this particular morphology with this particular intracellular junction being involved, right? The fact that this involves the desmosome, which attaches cells to its neighbors, means that the separation occurs between individual epithelial cells, right? That's the extent. In contrast,
Okay, thank you.
In contrast, if you look at the hemidesmosome, the disease we used to illustrate this is illustrated at the bottom there. This was bullous pemphigoid, right? So if we disable our hemidesmosomes on the basal surface of the cell, we get a separation between the basal membrane of the epithelial cell and the underlying basement membrane. And that means that the blister is subepidermal. It's beneath the entire thickness of the epidermis. But if you'll notice in this histology, the epidermis over the blister remains intact because there's no cleft within the epidermis. The cleft is below the epidermis. And that location is because of the location of the affected intercellular junction. Okay?
Thank you.
Don't forget to, this is a nice slide because you have to remember that when if we show you a transmission electron micrograph, know your layers, okay? Basal lamina and reticular lamina, right? And know your major proteins associated with the intercellular junctions, as well as the cytoskeletal filament component associated with those junctions. Okay, so that's intercellular junctions. Let's talk about apical specializations, okay? And we just focused on microvilli, cilia, and stereocilia. Those are the three. And the functions of these things is generally either to increase the membrane surface area to enhance absorption or to facilitate movement of material along an epithelial surface. So the first one is the microvilli, okay?
That's why we see a lot of these.
Right? So these are cytoplasmic projections, and we stress the fact that we generally can't see individual microvilli in light microscopic images. We have to look through an electron microscope to actually see the microvilli. What we see in the light microscope sections is the brush border. Okay? So brush border we visualize under the light microscope. Microvilli we visualize under the electron microscope. The drawing on this slide shows the green, you see the green bundles there? Those are actin filaments. So you should always remember that actin is the major constituent protein in the core of the microvilli, right? Microvilli are motile, so they actually also are myosin molecules, but the core major protein is actin, filamentous actin. The actin protein extend through the core of the microvilli and then insert in the apical cytoplasm into the terminal web, which is a web of actin filaments around the apical, just under the apical membrane of the epithelial cells. Okay? The size of the microvilli are about 1 micron in length and 1/10 of a micron in diameter. This is just a blow-up of that transmission electron microscope view, and you can see the arrow is pointing to a single microvillus, and you can see in the core it has like it has some lines in the core. These are the actin filaments. You can see them here, right? And this is the root or rootlet of the microvillus within the apical cytoplasm. And these fibers down here are the terminal web, okay? Does anybody remember what the G stands for here?
The glycocalyx?
Exactly. Thank you, Camilla. This is the glycocalyx, which sits on the top of the microvilli and is a sugar-rich area, right? It also contains digestive enzymes as well. So this is the structure, the ultrastructure of the microvillus. Now look at the inset. The inset is cross-section, the greater magnification of microvilli. The arrow is pointing to the actin filaments in the core. This inset is here to provide a contrast with the cross-sectional anatomy of the cilia. Okay, so you'll see we give you cross-sectional electron micrographic views of both, but they look quite different. Okay, this is a view of what the microvilli look like when you look at them under a light microscope. You can't visualize them. What you do see is a brush border. And that's this pink line on the apical surface of these intestinal epithelial cells. And this is a section of renal tubules, which shows a brush border at the apical edge of the renal tubular epithelial cells. Okay?
Hmm?
Yep. Was that a question or just a, that was nothing? Okay. Another thing to notice is you'll see sometimes in some of these sections, you can see a line at the periphery of an epithelial structure. At this renal tubule, this line at the outside is the basement membrane. Okay? Our second apical specialization is the cilia. So cilia differ from microvilli. Number one, you can see them on H&E stained light microscopic sections. Look at this image down here. The cilia are distinct and easily seen along the apical edge of the cell. So if we give you a picture of an epithelium and you can see these hair-like projections on the surface, they are not microvilli, okay? If you can see them like this and they're this distinct, they're either cilia or the other one we're going to show you in a minute, stereocilia. But cilia are much more distinct, actually, and I think, much easier to see than stereocilia, okay? So one of the key differences with cilia is, well, they are very motile. That is actually their major function. Remember, is propelling material along the epithelial surface. And they do that because their structure contains microtubules. It does not contain microfilaments. It is microtubules. They are much larger than microvilli. They are up to 10 microns in length, and they're twice the diameter, 2/10 of a micron in diameter. And they function to beat, and they exhibit a real beating pattern on the surface of the epithelium, which moves material along. Mariam, you have a question?
Yes, hi. I wanted to ask if we would need to know the specific like sizes, size difference between like the cilia and the microvilli.
Yeah, we're not gonna ask you how many microns it is in length. We're giving you this to emphasize the fact that it gives the apical specialization a very different appearance, because the cilia are 10 times the length, and that's why you can see them when you look at the light microscope. Okay, but I wouldn't get crazy about 2/10 of a micron.
Okay, thank you so much.
Okay. Now these things are motile, right? So let's take a look at why they're motile. This is a slide that shows, this is a very important slide, okay? Because you see this right here. We showed you the cross-section of the microvillus in the last slide, right? This is a diagram of the cross-section of the cilia. This is in particular the axoneme of the cilia. And that's this portion of the cilia right here. Know all the parts of this diagram. Okay? They are peripheral microtubular doublet. The little red thing sticking out is a dynean arm, which I think of as the motor of the cilium. The two circles in the center are two microtubules, and they're referred to as the central microtubules. The yellow spokes are called radial spokes, and then there's a link protein between the microtubular doublets that is a nexin link protein. So you should know those names and know how to identify them. You should identify them both in a drawing, but also in a transmission electron micrograph view. Okay? So if we show you a picture, a TEM, of a cross-section of a apical specialization, you should be able to identify whether it's a cilium or a microvillus, and then you should be able to identify the parts of that cross-section. Okay? The other piece of the cilium is when it gets into the cytoplasm, it is called the basal body. The basal body has a different ultrastructure. This is a drawing of the basal body ultrastructure. It's composed of microtubular triplets arranged in a pinwheel architectural pattern. Okay? So you should know that the axoneme, the portion of the cilia that extends above the apical membrane of the cell, is its internal structure is microtubules in a 9+2 assembly. And the structure of the basal body of the cilium is microtubular triplets arranged in a pinwheel formation. Okay? Oh, one thing, I think I pointed this out in class, but you'll notice that in this view of longitudinal sections of the cilia, that in this electron micrographic view, we can see microvilli. See, they're labeled MV, and look at how much smaller they are than the cilia. Okay? So the microvilli are just much smaller than the cilia. And the third and final apical specialization is the stereocilium. So these are much less common. They have a very limited distribution, male reproductive system and inner ear. Okay? You should be able to identify the type of epithelium. It's usually described as a pseudo-stratified columnar epithelium that has stereocilia as an apical specialization. A couple of contrasting features of stereocilia: they contain microfilaments that are active, just like the microvilli, but they're much longer than the microvilli, and they have a branched morphology. Remember, microvilli are just straight, but the stereocilia are longer and they have a branched morphology. And because they're longer, you can actually see them at high power on light microscopy. Okay, you don't have to look at them at an electron microscope. Stereocilia are longer than microvilli, but they're less motile. Okay. So let's talk about any questions about the apical specializations. Okay, let's move on to the types of epithelia. First, we're going to talk about lining or covering epithelia types. So we classify epithelia as, first of all, the number of cell layers. If it's a single layer, it is a simple epithelium. If it is two or more layers, it is a stratified epithelium. And then we further classify it in terms of the cell shape of the most superficial cell layer. So squamous cells are thin, flat cells. Cuboidal cells are cells who are about as wide as they are thick or tall. Columnar cells are taller than they are wide. So this chart from Junqueira basically lists all the different types of lining or covering epithelia and describes where you're going to find them, their distribution. And you should know something about this, okay? Something about where we're going to find each of these epithelia types, right? And then something about the function of the epithelium, which, as you'll notice, for the lining epithelium are usually quite similar. They usually have to do with protection or lubrication, right, of the cell surface. So you should know all these different types of epithelia. And you should also know the exceptions, which are the transitional or urothelial epithelia. And you should know the idea and the concept of pseudostratification, because we also talked about pseudo-stratified columnar epithelium. We've got some pictures to illustrate this, so I think this makes it easier. Examples of simple squamous epithelium. We're talking about this layer here. This is a blood vessel, okay? So this is a muscular artery, and we are just focusing on the inner lining, which is right here. You'll notice that is a single layer of cells. So this is a simple epithelium, and the cells are flattened, so they are squamous. This is another example, which is the mesothelial lining of a serous cavity. The serous cavities are pleural, peritoneal, and pericardial. So the lining of the cavity has a single cell layer lining of flattened cells. So this is also a simple squamous epithelium. Cuboidal epithelium. On the left, we've got an example of the lining of a renal collecting duct. And we're talking about this epithelium right here. This here is actually a blood vessel. It's a capillary. And this would be a simple squamous epithelial lining. But this is a simple cuboidal epithelial lining. And we also see a similar lining on the surface of the ovary. Okay, so these cells are about as tall as they are wide, so they're cuboidal. It's a single layer, so it's simple cuboidal epithelium. We also have simple columnar epithelium. On the left, we got an example from the gallbladder. See, we have one cell thick, but the cells are much taller than they are wide. So this is a simple columnar epithelium. Similar situation in the small intestine. We have one cell layer thick. The cells are taller than they are wide. Another example of simple columnar epithelium. Now, moving on to the squamous epithelium. This is a section of skin, which is the one place that we often see keratinized, stratified squamous epithelium. I think we have two here. Emma, got a hand raised.
Yes. Would you agree that simple epithelium tends to be in areas where you need like faster exchange of material or like absorption of things in the lumen, or because I know that's with the blood vessels specifically.
Yes, I would agree. Remember, form often follows function. So there's often a functional reason why the cells that we're studying and their arrangement in tissues look the way they do. And it is true that simple squamous epithelia provide a thin layer that has to be transgressed by fluids, electrolytes, or macromolecules. So yeah, and that's one of the reasons why endothelium, the simple squamous epithelium lining blood vessels, is a single cell thick, right?
Thank you.
Zachary?
Yeah, hi. In the last slide, why is the epithelium on the right, in the picture on the right, why is that not pseudostratified columnar epithelium?
It's very much in the eye of the beholder. Now, since I've been looking at these things for a long time, I will tell you that I happen to know that the small intestinal epithelium has a native population or a usual population of small lymphocytes. Some of the nuclei that you're seeing here are lymphocytes. This, right? The epithelium are basically pretty much a single layer, and they're pretty much at the same level, though I have to admit that sometimes our descriptive things in real life don't always match. So you can make an argue that some of these things are at different levels, but when you look at your pictures of actually pseudostratified epithelium, they're at very different levels. So they go all the way down to the basement membrane and then they come all the way up. You see, these are really pretty much in the same central zone of the columnar cell. Okay?
And what about the goblet cell portion? Although I guess those are...
Yep, goblet cells, that's interesting because goblet cells are an example of a glandular epithelial cell. They're a unicellular exocrine gland scattered in the midst of a lining epithelium. Glandular cells can either form an entire epithelium or they can be scattered as individual cells within another epithelium. And the latter case is the situation with the small intestine.
So in the first case, that would be pseudostratified, where it spans the whole, where the goblet cells, which would span the entire pseudostratified epithelium. And here, where
I'm not sure what the first case is.
No, sorry, because you mentioned two cases where one is where it's scattered and one is where it spans the whole epithelium.
No.
So
no, when I'm talking about scattered, I'm talking about scattered along the epithelium, like that, not scattered in terms of level in the epithelium. So if you look at small intestinal epithelium, you'll see goblet cell, absorptive cell, absorptive cell, absorptive cell, absorptive cell, goblet cell, right? I'm not talking about scattered in the thickness of the epithelium.
Right.
So,
And so comparatively in the pseudostratified, it would be spanning the entire epithelium, right? Like instead of it being goblet cell, absorptive cell, goblet cell, it would be like constant goblet cell, right? Or am I confused on that?
well, goblet cells, remember, goblet cells are a glandular cell that is usually an individual cell within a lining or covering epithelium, right? So we don't usually have an epithelium that's composed entirely of goblet cells, okay?
Okay. Okay.
Maybe
I understand.
in a couple slides, we'll talk about the goblet cells, and maybe then it will begin to make a little bit of sense. To share, you got a
Okay.
question?
Yeah, can you go to the previous slide
Yep.
with the simple, oh wait, the one before actually.
Yep.
Yeah, can you explain? So the left, I think I'm having a bit of a hard time just visualizing how that's simple because that looks like multiple layers to me.
Okay. So you always have to identify what cell you're looking at, okay? This is a section of a muscular artery. Most of these nuclei are in the section of the artery, which we'll get to later on in the year, that is called the media. This is the muscle coat. All of these cells are smooth muscle cells. We're not talking about them. We're just
Okay.
talking about the epithelial line, and that is only these cells that line the
Okay.
structure.
Okay. The apical cells
Yeah, I mean you
are
can.
apical to the lumen.
Okay. Don't use the word apical here. They are
Okay.
adjacent to the lumen. When we say apical
Adjacent.
basal, we're talking about parts of the epithelial cell.
Okay. Oh,
What I'm
gotcha.
talking
Okay.
about here is what are the cells that are actually lining this space, this lining epithelium. Okay?
Okay. And you like for me to look at this photo,
Yeah.
I can't just say, oh, like the more pink part,
Yeah.
the cells look like more stretched. They don't even look, I mean, they're
Right.
flattened, but it doesn't look
Yeah.
like the cells up top. Okay.
Yes, very good. They don't. And
Okay.
this is because, and in a couple of weeks, you'll see why they don't. These are connective tissue cells. So they're elongated and they're different. They're a different type of tissue than we're talking about this week, which is the epithelium.
Perfect. Thank you.
Good. Despina.
Hi. I was wondering if, like, on one of the last slides where we were looking at, I think the gallbladder and something else, are we going to be, I understand that we're expected to identify that this is a simple columnar epithelium. Are
Right.
we also expected to be able to say, okay, this was obtained from the gallbladder?
No. I mean, basically, at this point, we're focusing on the types of epithelium. In a question, I mean, there might be something about where you find these things that could be like for instance, when we were talking about stratified squamous epithelium, we make a distinction between keratinized and non-keratinized. So we would expect maybe you would be aware that keratinized is usually skin for the most part, but the
Yeah, as
non-keratinized
opposed to like the
occur
esophagus,
in
for
all these
example.
other locations.
Okay,
So that's kind of the level. But we're not going to show you a picture and say, what organ is this from?
great. Okay, thank you so much.
Okay, Mariam.
Yes, hi. We can go back to like the slide that we were on, like the keratinized epithelium.
Okay.
I'm having trouble understanding or being able to differentiate what keratinized versus non-keratinized looks like.
Sure. Yep. Okay. So let's look at this picture first, okay? We're going to talk about this in 2 weeks when we're talking about skin, okay? But stratified squamous epithelium, particularly in the epidermis, matures as it progresses from the basal layer to the surface. The most differentiated cells are these in the superficial part. And the most differentiated cells, this is the keratin. At the most superficial layer, these epithelial cells no longer have a nucleus and are basically just a bag of keratin. So when you look at the section, keratinized epithelium has layers of anucleate pink material like this that looks kind of fibrillar and follows the surface of the epithelium. In contrast, this is non-keratinized. you can see cells with nuclei all the way up to the surface of the epithelium. But we don't have that layer that is anucleate with deep red or deep pink cytoplasm, which is the keratin. Okay, so if you go back and forth, you see the keratin's here and no keratin here. So this is keratinized. This is non-keratinized. Okay?
That makes sense. Thank you so much.
All right, and look here. See, the other thing is we're pointing out that the keratinized most commonly is skin, non-keratinized esophagus, oral cavity, vagina, and ectocervix. Okay, so basically, moist squamous mucous membranes are generally non-keratinized. Skin is generally keratinized. Okay.
Thank you.
How about this one? Olivia. Olivia, did you have a question?
I did, just a quick one. So on an exam, you had mentioned that the importance of this table of the lining of examples of distribution.
Yeah.
So potentially on an exam, you could like give us a cell type and say, this is from the gallbladder. What type of cell type is this? So that would be, is that like an example of a question you think? Or no?
Yeah, I guess we could. We're not going to be emphasizing your ability to identify the location of an epithelia that we show you. But you should know in general terms where you're likely to find these different types. Like you should know that keratinized squamous epithelium is skin. And you should know that these mucous membranes that we mentioned are non-keratinized, stratified squamous, right? So, but that's kind of the level. Like I would know, we're going to show you ciliated columnar pseudostratified epithelium. That is a classic that you should know is usually associated with the respiratory tract.
Okay.
Okay, that kind of level, right?
Thank you.
This one here is an unusual one. Okay, so this is stratified cuboidal, only seen in a few locations. Sweat glands is a big one. Okay, so and you'll notice that this is stratified because there are two layers, right? And it's cuboidal because the cells are about as tall as they are wide. Oh, here is the classic, right? So if you see this picture, this is ciliated pseudostratified columnar epithelium. Now, Zachary was talking about the idea of pseudostratification. Look at this. See the different levels that the nuclei are at? It looks like these cells are piling up on each other. But in fact, there's a drawing in the, I think in the lab PowerPoint that shows that these cells actually have different layers. They all go from the surface all the way down to the basement membrane. It's just that the nucleus is at a different level. So it's not a truly stratified epithelium. It looks like a stratified epithelium. Does that make sense? So pseudostratified ciliated columnar is very characteristic of the respiratory tract, particularly the trachea and the bronchi. Okay? Let's see. Is it Kinneret? Kinneret?
Sorry, yeah.
Yep. Is that how you pronounce your name? Kinneret?
Kinneret, yeah.
Yeah. Okay. You have a question?
Yeah, so with this one, is it not stereocilia or is that considered the same just when it's called ciliated?
No. Okay. So cilia, there are different types of cilia, and you should be able to identify whether it's cilia, which some people refer to as motile cilia, because that's one of their major functions, or stereocilia. I'm going to show you a picture of the epididymis, and it's from the lab PowerPoints, I think. Look at that picture. You'll see it looks very different. So yes, I would expect you to be able to look at those pictures and say, this is cilia like we see in the respiratory tract, and this is stereocilia. Okay?
Okay, thank you.
By the way, you'll notice that the arrows point to goblet cells. So this is an example of a lining epithelium in which there are scattered unicellular glandular cells, the goblet cells. Okay? Up, here is the picture. Can you see it? This is the picture.
Yeah, so much longer.
Spermatozoa here. Now look at this high-power view of the epithelium. There are hair-like extensions at the apical surface of the cell, but they look different than the cilia we just saw in the respiratory tract. Okay? The reason they do is because they're kind of long and they're branched. So when you look at them under the microscope, it has a different appearance than the cilia we see in the respiratory tract.
Okay, that makes sense. Thank you.
Okay. Here's one of our exceptions. Oh, Gareth Lynn, there you are. You have a question?
Sorry, I did have a question. Because I know the one that you just described was the.
I'm having trouble hearing you. Can you turn your volume up? Oh, now I think you're muted. Try speaking again.
Okay, can you hear me
Yeah,
now?
okay. Yep.
Alright, so I'm sorry, it's probably gonna sound a little redundant, but I just wanted to specify the last issue that you just showed, not that one, it was on the pseudo.
The one with the pseudo-stratified?
Yeah, that one.
Yeah, okay.
How is that one in particular different from the one that was shown earlier that was described as the simple columnar epithelium that was in the small intestines? Because you can see like the goblet cells, like
Right.
the nuclei don't look uniform, so it looks pseudo-stratified in a sense. You know what I mean? So between
Yeah,
those
okay.
two, like
So yeah, the
makes
intestine, right, the intestine has some similarities, okay? So one of the differences is if you look at small intestinal epithelium, you will not see these hair-like things on the apical surface. You'll see a pink line, which is the brush border under the light microscope. Because in the intestinal epithelium, the apical specialization is microvilli. In this ciliated pseudostratified columnar epithelium, the apical specialization is cilia. So they look different. You're absolutely right to point out that both of these epithelia have scattered goblet cells. So at first glance, they may look similar, but you have to look for the other clues, right? The apical specialization and the fact that the respiratory epithelium is really pseudostratified. You've got nuclei at different levels of the cell. Whereas in the intestine, they tend to be in the middle of the height of the cell, in the middle zone. Okay? Okay. This one, urothelium. You know, the key feature of urothelium to look for is this umbrella cells. You see these dome-shaped cells on the surface. That is very characteristic of the urothelium mucosa, also called the transitional mucosa. Okay? And one thing about urothelium is that when the bladder is distended, full of urine, the urothelium stretches, so it becomes thinner. So it may only be 1 or 2 cells thick in a distended bladder, but in an empty bladder, it's going to be usually 5, 6, or 7 cells thick. Okay? Okay, next topic we're going to talk about. Any questions about the types of lining epithelium? Okay. Leah.
For the urothelia, urinary bladder, I read some information about it being like more than the urinary bladder, like the whole urinary system. Is that true?
Yes, that's true. It is more, it lines the bladder, but also the ureters, the renal pelvis, and also a portion of the urethra.
Perfect. Thank you for the.
Types of epithelia, secretory epithelium. So we've been talking about lining epithelia, and now we're going to talk about secretory epithelia. Okay, these are epithelial cells that function to produce and secrete various macromolecules. And here's the part that that Zachary was getting at before. May occur in epithelia with other major functions. An example of that is the small intestinal epithelium, where the major function is absorption, but we have scattered goblet cells. So that is individual cells appearing within that epithelium. Or these secretory epithelial cells can be all of the cell type that's present in a gland. Make sense? So if you look at an example of a gland like the adrenal cortex, all of the cells, all the parenchymal cells in the gland are glandular cells, right? If we look at the epithelium in the small intestine, only a minor number of the cells in the epithelium are glandular. Most of those cells are lining epithelia that are specialized for absorption. Okay? Secretory cells synthesize, store, and release proteins An example would be the pancreatic acinar cells. Lipids, like we see in the adrenal gland or the sebaceous glands of the skin, or carbohydrates and proteins, like we may see in salivary glands. The mammary glands secrete proteins, lipids, and carbohydrates. And so they secrete all three of those types of substances. And the sweat glands don't synthesize much, but they mostly function to alter the water and electrolyte content of sweat. So they can make it more hypertonic or hypotonic, but they're not actually producing a secretion otherwise. Okay? Now here's the cell that we've been talking about, the goblet cell. So goblet cells are sometimes characterized as unicellular exocrine glands. Okay? And they're very commonly scattered in other types of epithelia like simple cuboidal, simple columnar, or pseudostratified. So the simple columnar example would be the small intestine, and the pseudostratified example would be the respiratory epithelium. Both of those epithelia have scattered goblet cells. Okay, the image shows that the goblet cell produces mucin within the vacuole and then secretes it onto the surface where it becomes mucus. So mucus is just hydrated mucin. Okay. This is a transmission electron micrograph that shows the secretory goblet here, and then the nucleus of the goblet cell here. Okay. How do we classify glands? Now we're talking about exocrine glands. Okay. We split glands into exocrine glands, which are glands that produce and deliver a secretion through a duct system, and endocrine glands, which are glands that produce a secretion that they secrete into the bloodstream. Okay? We're focusing here on exocrine glands. We classify exocrine glands as simple or compound based on the structure of their duct. Let me just see. So glands have both a secretory portion where the secretion is produced and a conducting portion, which is the duct that delivers the secretion. So we classify the gland on the basis of whether its conducting portion is simple or straight, or divided, split. Okay. We further classify the gland in terms of the appearance of its secretory portion. So in the top row, we've got the simple glands. They all have a single straight duct. And we have various different morphologies of the secretory portion, the deeper portion of the gland. It can be straight. This would be an example you would see in the small intestine, for instance. There's a straight, simple tubular gland. It can be branched. It can be coiled tubular, like we see in the sweat glands, or the secretory portion can be arranged as a little pouch at the end of the duct, which is usually referred to as acinar or alveolar. The more complex or compound glands have split or forked ducts. And you can see that we then classify them further in terms of what the architecture of their secretory portion is. Is the secretory portion straight tubules? Is it little pouches? Or is it both, tubules and pouches? So we call it tubular acinar or tubuloacinar. Okay? You should know the different mechanisms that these glands, the exocrine glands, use to secrete their product. So there are three different mechanisms: merocrine, holocrine, apocrine. Okay? Merocrine is the most common, and this is the method that is used when glands, usually glands producing a protein secretion, secrete their product into the lumen of the duct. This is illustrated here in this drawing. And so this works by exocytosis at the apical end of the secretory cells. And this is an important feature without loss of cytoplasm. So this is something that distinguishes merocrine secretion from holocrine secretion or apocrine secretion. In merocrine secretion, we have a secretory vacuole that travels to the apical cell membrane, It fuses with the apical cell membrane and then extrudes its contents onto the surface. We don't lose any cytoplasm or membrane into the secretion. Okay? In contrast, the other two methods do lose membrane and cytoplasm into the secretion. My favorite is the middle one, hologram secretion, because this is really only seen in sebaceous glands or follicles. And in this method, the cell blows up. Okay, the entire cell fragments, and that becomes a secretion. So it's ba-boom, the cell explodes, and that's your secretion. Okay, sebaceous glands of the skin only. Apocrine secretion on the right side is a method in which the secretion accumulates at the apical side of the cell, and then the apical cell membrane and the cytoplasm pinches off and is released into the lumen. Okay, and we see this when there's a lipid-rich secretion involved. Okay, now the breast gland, the mammary gland, produces breast milk, which is a combination of lipid, protein, and carbs, right? And breast uses both merocrine secretion and apocrine secretion as its method of secretion. The proteinaceous component is secreted via merocrine secretion, and the lipid-rich component is secreted via apocrine secretion. Okay? Okay, pemphigus vulgaris, we already saw this, right? Remember we talked about...
Sorry, I just had a quick question.
Yep.
For the slide on the acinar and tubular acinar, I just had a question that they look so similar, those
They
two.
do.
Like, what's the real, like,
Yeah,
are they kind of overlapping? Like, how does that?
they overlap like crazy. I have trouble identifying this on slides. I think this is sort of like a theoretical construct. Certain, you could learn, looking at the bottom, the examples. You can see examples of where these things are seen, but we're not going to focus on, is this an example of a compound tubuloacinar gland or a compound acinar gland? That's too much. I mean, I can't do that. I don't expect
Okay.
you to do that.
Thank
But
you. That makes sense.
you should just understand something about the basic way that these exocrine glands are classified based on this morphology. Okay?
Okay, thank you.
Mariam?
Yes, hi. Can you give us like an example of a type of question that would be asked about these glands?
Well, you might have a description of a gland. For instance, you might have a description, say, of a sweat gland, in which part of the description might include the fact that it is a coiled tubular gland. But if we do that, we're going to give you other information as well. Like the gland is involved in, I don't know, controlling the temperature of the body, and the glands are all over the place in the skin, stuff like that. There should be other clues, and we're not going to rely on you actually just solely relying on this kind of architectural classification.
Okay, thank you.
Okay, now let's see. I think, did we discuss this? Of course, is pemphigus vulgaris. I think we already actually discussed this. Okay, so that's pretty much what I have. Does anybody have questions about anything before I move on to week 1?
Could you quickly go back to the slide with so I think it was microvilli and cilia. I just wanted to take a picture of it. I didn't get it.
Let's see. Well, this is a good one of cilia.
I think it was this. I think it might have been this one.
This one you like? Okay.
I think it was this slide. Thank you. I just didn't grab a screenshot of it. Thank you.
Sure. Olivia, you have a question or no?
Yeah, can you hear me?
Yep.
So for the cilia, the cross section, where you emphasize the importance of knowing like the different parts.
Yep.
Can you show on the cross section from the TEM where you would see like the arm and...
Yes. Let's see. So here's the cross section, right?
Yeah.
Now, this cross section, I can't see all the parts. Okay, so what I can see on this cross section, I can see the peripheral microtubular doublets. That's here, here, right? I can see the central microtubules. There's 2 in the middle. I can see some of the radial spoke. See that? What I can't quite, I think I can see a Dinean arm on this one. You see that? They're little nubs that kind of stick out on the side. And I can't really see the Nexin linking protein. If we give you a question like this, you'd have to be able to see more detail than this. That's why I think you're okay. If you just learn the diagram, you should be. I couldn't show you this picture and put an arrow on something and say that's the Dinean arm. It would have to be a bigger image that you could actually see it. Okay? But I did pretty good. I identified most of them. Okay? And Gina.
Hi, good afternoon. I just wanted the same slide. So I know like for the cilia and the flagella, they both have like the 9+2 arrangement.
Yes.
You guys wouldn't give like a TEM of like this picture and like ask between the two, because how would you distinguish between the flagella and the cilia for like this image?
You are way ahead of us. You are way ahead of us. We're not going to ask you anything about flagella. But you're right to point out, the spermatozoa have a very similar structure to this, but it's not something we're going to focus on for this at this point in this unit. So just learn the cilia and the
Thank you.
Okay, Majid.
Hi, good afternoon. I just wanted to confirm for the axoneme, it's microtubular triplets, not doublets, correct?
No, the axoneme is microtubular doublets in a 9+2 configuration. The basal body is
the base. Okay,
triplets in a pinwheel formation.
gotcha. Thank you very much.
I'm glad you asked that. Anybody else? Is Dr. Brown in the house?
I'm here.
He's here. Okay.
Okay, so I'll continue.
I'm going to stop share, right?
All right.
Stop share. Okay.
All right. So I have to put my share on here. Let's see if I can do that.
Yep.
Says here, send request for sharing. I guess I have to send a request.
You're supposed. Okay, send a
I think
request.
you have to transfer something
Okay,
to me.
you are supposed to be a. You're supposed to be also a host, but okay. Can you send a request? I'm looking for it.
I don't know. There it goes. I've sent a request.
There it is. Abron 6 is requesting. I'm going to allow that.
That's good. That's
There
good.
you
Makes
go.
things.
Yep.
I
try to be cooperative.
At the end, I think I have to send you something to revert the power back to you.
Okay.
So that the recording could be sent out.
Oh, I think I'm going to get it anyway. Okay.
I don't know if it goes out to me or to you, but Okay.
We can ask these guys for help if we have.
Yes. I'll probably need some help. Okay, so let's get started here. Let me see if I could share my PowerPoint. I have to select the correct PowerPoint number. This one I think is the one. Let's share that. And do you see my PowerPoint?
Yep.
Yes.
You see a full screen PowerPoint or not?
Yes.
You see a full screen? Okay. Yeah, okay, that's what I intend. Let me try one thing. It's a little easier for me to have it here where I look at the screen I'm looking at shows my image here. Let's see if this works. How's that? Okay, you see me and you see my full screen, correct?
Yes.
Yeah.
Okay. All right, let's get started. Let's see if this works. Hopefully it works. Okay, so I'm talking about the first week. We covered several things together. We covered histologic methods. Basically, I want to talk about briefly the routine stain that we do. It's called hematoxin and eosin. It demonstrates certain principles about staining that you need to know when we show you slides. They're usually H&E, hematoxin and eosin, which is a mixture of two dyes, an acidic stain and a basic stain. So it gives you much more information than just using one single dye. If you simply used hematoxin alone, everything would look blue. You see my arrow when I point to that?
Yes.
Okay. If we just use eosin and we look something like this, you just see pink. If you use both together, you see both blue and pink together. Well, conveniently, blue and pink don't seem to stay so much the same structure, although they might. Often it's different structures which stain. So the nuclei tend to stain blue with hematoxin. The nuclei are basophilic. They have an affinity for the basic stain in this routine stain, which is hematoxylin. The pink structures pretty much are cytoplasm, mostly cytoplasm, which stain with eosinophil. They stain pink with eosin. So eosin is the acidic stain and cytoplasm is the acidophilic structure. And together you get blue nuclei and pink cytoplasm. Now, you could apply this to any tissue at all. If you stain muscle, for example, the cytoplasm muscle tends to stain very nicely with eosin, whereas the nuclei continue to stain blue. If you look at red blood cells don't, mature ones at least, don't have nuclei. They simply are bags of cytoplasm. So there the cytoplasm stains pink. If you look at extracellular matrix, the extracellular matrix has lots of collagen in it. Collagen stains pink. Collagen has an affinity for the acidic stain, which is eosin. So collagen is acidophilic and stains well with eosin. I don't really have that to show you easily here, although I do see some blood vessels which have some collagen in the wall, and you see the vessels here tend to stain somewhat pink, partly reflecting the collagen in the wall of vessels. So in general, if we want to generalize, hematoxylin, the basic stain tends to stain nucleic acids. DNA is a nucleic acid. RNA is nucleic acid. So cytoplasm, although it usually stains quite well with eosin, if there's lots of ribosomes, which is mostly nucleic acid, in the cytoplasm, the ribosomes will stain rather bluish. Actually, if you look at this picture here of, this happens to be pancreas, there is a little bluishness in the cytoplasm, like right here, for example. That's because there are areas with lots of rough endoplasmic reticulum. The nuclei certainly stain blue, but there is some bluish cast to some of the cytoplasm where there's lots of rough endoplasmic reticulum. So one problem with looking at slides is simply to recognize different stains. There are a variety of special stains. I'm not talking too much about them today, but H&E is the workhorse stain. Another problem is simply orientation, and that's something you gain experience with as you look at slides. When you study standardized or idealized planes of section, it's easy to recognize tissues that way, but in reality, tissues can be cut in any plane just by gaining practice, by looking at things and reconstructing in your mind the structures. You can figure out the structure of tissues. You could do this in your spare time on real life items like an egg, for example. If you're having a hard-boiled egg for dinner, hopefully you have more than that, you could cut it in various planes, and the same egg looks different depending on the plane you cut it. If you're having a lemon for dinner, I hope you have more than that too. You could cut it in different planes, and the lemon looks quite different depending what plane you cut it at. Well, the same is true for tissue sections. You can take a tube, for example, a blood vessel, like we see here on the right-hand side, or an intestine, from a small animal, of course, and you cut it different ways. It looks radically different depending how you cut it. It's not so hard to learn to reposition these things in your own mind and see things and understand things, even though they're not necessarily always in an idealized plane, like a coronal plane or a sagittal plane or a transverse plane. Okay, let's talk about cell membrane and cytoplasm. Cell membranes are ubiquitous structures, just like cytoplasm, and basically they consist of two lipid leaflets. Here you see two leaflets, which are predominantly made of phospholipids. Here you see a layer of phospholipids on one side, and here you see a layer of phospholipids on the other side. This side faces the extracellular environment. This side faces the cytoplasm. And if you look closely at a phospholipid, it's actually a amphipathic molecule. It has a polar head like this right here, and a hydro or nonpolar or hydrophobic tail, like you see here. The hydrophobic tail consists of fatty acid chains, like you see here, and the polar head consists of several different small molecules. Because of the bipolar aspect of phospholipids, with the polar aspect on one side and the hydrophobic tail on the other side, they arrange themselves in two layers. Because of that construction of phospholipids, they arrange themselves in two layers. That accounts for this two leaflet structure of a phospholipid-based membrane like we see in cells. And because there are fatty acid tails, which are nonpolar, in the center of the membrane becomes a barrier to polar molecules or ions. The membrane is actually a barrier, a physical barrier. Now, embedded within the membrane, which is basically composed of phospholipids, are a number of different proteins. And these proteins give properties to the membrane, which it would otherwise lack. Some of these proteins form receptors for molecules. Some of the proteins form transport molecules or make channels or for movement of molecules from one side of the membrane to the other side of the membrane, for example, from the extracellular space to the cytoplasm or from the cytoplasm to the extracellular space. Some of these protein molecules are important for maintaining an electrochemical gradient across the membrane, so there may be more negative charges on one side and more positive charges on the other side. This is important for the behavior of the cell. For example, in cell signaling, it's an important feature. On the left-hand side of this diagram, you see a listing of some of the functions of the cell membrane, which is often called the plasmalemma. And to a large extent, that reflects the structure of the membrane. Actually, you could think of the membrane as consisting of a semi-fluid lipid layer, the lipids, into which are embedded a mosaic of proteins. Sometimes it's called the lipid mosaic model of a cell membrane, to have those two different constituents. If you look carefully at this cell membrane here on the right-hand side, you see some of the proteins actually are inserted deeply within the membrane and travel all the way through the membrane like this. Other proteins are just lightly opposed or attached to the surface of the membrane. Here's a protein which is lightly attached to the surface, like right here, for example, maybe over here and over here and so on. So some proteins are intrinsic. They actually are embedded deeply in the membrane. Other proteins are more superficial. They attach lightly to the surface of the membrane. Now, one of the functions of membrane is to form a semi-permeable barrier, but there must be a way to carry molecules from the outside to the inside and the inside to the outside. There must be a way to allow membrane transport of needed materials and membrane transport actually of waste products, which a cell has to get rid of. So here's a diagram from Junqueira showing you several ways that cells move things across the cell membrane, which, as you know, is a semi-permeable barrier. There is non-vesicular transport, where a little vesicle does not butt off the membrane in order to transport materials. And here we see there are several different varieties of non-vesicular transport. There's some molecules which are totally nonpolar, for example, oxygen or nitrogen, may simply diffuse through the membrane because there is a lipid barrier within the membrane, but if a molecule is totally nonpolar, it may still diffuse right across the membrane because it's nonpolar. You see it demonstrated right here. Other molecules are polar or ionized, and they may travel through the membrane via a channel. So if a protein makes a channel like we see here in B, molecules that are polar or ions may actually be able to move through the pore from one side to the other because now there's a space which has water within it, and the polar molecules or ionized substances can move right through that water channel. And finally, there are proteins which are carriers or transporters. They bind to a substance on one side, and they they change conformation, and they move the substance into the to the other side of the membrane. So, if this is outside the cell here, the substance here binds. There's a change in conformation of the carrier transporter, and the molecule or substance is moved to the inside. So these are three ways in which materials can move from one side of the membrane to the other. Now, in addition to having these non-vesicular means of transporting materials from one side of the membrane to the other, there are vesicular methods of transporting materials. In other words, a small bubble or balloon can be made from the membrane. And doing that, materials can be transported from one side of the membrane to the other. So here we see pinocytosis, which is a form of cell drinking. But fluid and maybe some ions or small molecules dissolved within that fluid can be imbibed into the cell when a bit of membrane pinches off from the surface. You see here how the membrane is closing up on itself to form a vesicle, and then it pinches off totally to form a self-contained vesicle now released into the cytoplasm. So that form of taking in materials, fluid, and some dissolved materials within the fluid is called pinocytosis, cell drinking. Alternately, there could be small pieces of particulate matter, even things like bacteria or cell debris from dead cells, that could be taken into the cell by a more, let's say, aggressive form of vesicle formation. That's called phagocytosis. The vesicle which pinches off from the surface, in this case, might be called a phagosome. Okay, so that's, you might call, to distinguish it from pinocytosis, you might call that cell eating, a form of cell eating. So pinocytosis, cell drinking; phagocytosis, cell eating, even whole bacteria and particulate cell debris. And then there's a more selective form of internalizing materials within vesicles that's called receptor-mediated endocytosis. So there may be a receptor there for something that's in the interstitium here, and that something could bind to the receptor and then be internalized into the cell cytoplasm via a vesicle made of cell membrane. Again, by pinching off of a whole vesicle of cell membrane. Altogether, these forms of taking vesicles and their contents into the cell are called endocytosis. Those are all forms of endocytosis. On the other hand, materials can be secreted from the cytoplasm into the environment, into the interstitium, or onto a surface. That's called exocytosis. So endocytosis and exocytosis are, in a sense, opposites to each other. One is taking in materials, the other is exporting materials to the outside of the cell. And they actually balance each other. As vesicles come into the cell, membranes are brought back into the cell. As things are exported from the cell, membranes are added to the cell membrane. By balancing endocytosis and exocytosis, the cell membrane surface area remains the same, and the cell volume remains the same. So it's important that these things remain balanced to maintain cell volume. Okay, let's talk about organelles. There's a variety of different organelles. Of course, there's endoplasmic reticulum. Endoplasmic reticulum tend to consist, much of it actually consists of these flattened cisternae like this. These are flattened. You could take a vesicle in a way, or a sac, and flatten it out. And it looks something like you see here in this picture. That flattened sac is sometimes called a cisternae. Here you see it's labeled as a cisternae, but really it's a flattened sac. And these flattened sacs are in communication with each other. They are not separate sacs, really. They are in communication with each other. And often they occupy much of the volume of the cytoplasm. They often will extend all the way from the nucleus to the cytoplasmic membrane. So some cells have, relatively small amounts of rough endoplasmic reticulum. Other cells have lots of rough endoplasmic reticulum. It all depends on the function and behavior and metabolic uh, needs of that particular cell. It depends on the cell type. Now, rough endoplasmic reticulum is rough because on the surface of these flattened sacs or cisternae are many ribosomes. Ribosomes are particulate in their appearance. They look like small granules on electron microscopy. They're too small to see on light microscopy, but you can see them quite nicely on electron microscopy, and that's what you're seeing right here, rough endoplasmic reticulum. Now, and you see that here in the diagram as well. Now, rough endoplasmic reticulum is not the only rough endoplasmic reticulum in a cell. There's also smooth endoplasmic reticulum, which is very similar to rough endoplasmic reticulum, but it lacks ribosomes on the surface. So here you see in the diagram the smooth endoplasmic reticulum. It lacks ribosomes on the surface. They look different, and actually they don't quite have the same function. So the appearance, the structure, the morphology reflects a difference in function. They may be in communication with each other, but they have different functions. If you look at the actual electron micrograph here, if you look at the smooth endoplasmic reticulum, it does look smooth. It lacks ribosomes on the surface, but also it looks a little bit less like flattened sacs. It looks a bit more tubular. So if you look here, to me, it looks somewhat like anastomosing tubules, the smooth endoplasmic reticulum, in contrast to the flattened cisternae of rough endoplasmic reticulum. And actually, they look like they're in continuity over here. If you look carefully, it does look like they're in continuity. One thing about the rough endoplasmic reticulum, it actually is in communication or in continuity with the outside of the nuclear membrane. So the outer surface of the nuclear membrane, the membrane around the nucleus, which is a bilaminar membrane, by the way, it's a bilaminar structure of the nuclear membrane. Two membranes. By that, I mean two membranes separated by a space. When I spoke about the surface membrane, the plasma membrane, that is bilaminar, but in a different sense, in the sense that it has two layers of phospholipids binding to each other. When I speak about the two layers of the nuclear membrane, there are two separate membranes separated by an intermembrane space. You do not see that easily in this particular diagram, though. You're not seeing that in this diagram. Anyway, the rough endoplasmic reticulum is in continuity with the outer membrane of the bilayered nuclear membrane. Here's a higher magnification view showing you several flattened cisternae of rough endoplasmic reticulum. And again, we see lots of ribosomes on the surface of the rough endoplasmic reticulum. But in addition to that, we see some ribosomes which seem to be free. They are not attached to the surface of the rough endoplasmic reticulum. Here's an arrow pointing towards a grouping of several free ribosomes. These free ribosomes are sometimes called polyribosomes because they are actually all linked together by a mRNA. There's an mRNA which is helping in the process of protein production. It's helping produce protein. So the ribosomes are actually moving along the mRNA in the process of producing protein. And because of that, because there's a series of ribosomes moving along a single piece of mRNA, they all seem to cluster together. That's why they seem to cluster together. And sometimes they even form spirals or whorls of ribosomes because the mRNA in the center of that little assembly is holding them together in that configuration. So if you look at this, I see a little cluster of ribosomes. I presume they're all attached to the same mRNA. And here again, another cluster of ribosomes. I presume they are all attached on the same mRNA. So this cluster is called a polyribosome. That's a polyribosome. And here's a polyribosome, and here's a polyribosome. Now, these polyribosomes, some of them become attached to the surface of the rough endoplasmic reticulum. That's what we're seeing here, these attached polyribosomes with a bunch of with an mRNA synthesizing peptide and a clustering of ribosomes is what's attaching to the surface of the endoplasmic reticulum here. Okay, so here again we see rough endoplasmic reticulum on the left lower picture. Here we see smooth endoplasmic reticulum in the right upper picture. And here we're emphasizing the fact that these have different functions, although they look somewhat similar and have certain commonalities, they have different functions. So rough endoplasmic reticulum, important for synthesizing membrane packaged proteins. That is, proteins that are made for secretion, proteins that are delivered to lysosomes that form lysosomes, which degrade materials in the cytoplasm. They're the garbage disposals of the cytoplasm, and proteins that become incorporated into membranes. If you're wondering where the proteins which get incorporated into the various cellular membranes, well, they're synthesized in the rough endoplasmic reticulum. Okay. As far as smooth endoplasmic reticulum, SER, what does SER do? Well, it's important for some lipid synthesis. Steroid hormone synthesis occurs within smooth endoplasmic reticulum. Drug detoxification occurs in smooth endoplasmic reticulum, especially the liver. It could occur in other organs, but especially the liver, drug detoxification. And this also means not just illicit drugs, although, of course, that does reflect illicit drug use, but, drugs that people take for medication purposes, for health purposes. So those foreign substances, after all, that's what drugs pretty much are, need to be detoxified in some way. We can't have them in our body for any extended period of time. That occur, much of that, not all of it, but much of it will occur in smooth endoplasmic reticulum. In fact, as it occurs, if someone is repeatedly using the same medication or taking the same illicit drug, the smooth endoplasmic reticulum may increase in amount, increase in volume. Instead of a few tubules of smooth endoplasmic reticulum, you may have many, many tubules of smooth endoplasmic reticulum. So if a cell needs to have lots of smooth endoplasmic reticulum for detoxification purposes, you'll see a lot more than usual. If a cell needs to make steroid hormones, there's certain, like adrenal gland, for example, adrenal cortex. If it's a place where steroid hormones need to be synthesized, there will be a lot more smooth endoplasmic reticulum. And finally, there's a specialized form of smooth endoplasmic reticulum found in muscle, for example, skeletal muscle, which takes up and then releases calcium, which is important in the process of muscle contraction. This constant taking up and this cycle actually of taking up and releasing calcium involves the smooth endoplasmic reticulum, which surrounds the muscle filaments within the cytoplasm. So smooth endoplasmic reticulum in the specialized form of cells called muscle is important for storing, at least temporarily, calcium. So as you can see, there's a variety of different functions for smooth endoplasmic reticulum. This figure here summarizes a number of things I just mentioned to you. Rough endoplasmic reticulum, you have polyribosomes bound to the surface of the endoplasmic reticulum, and the peptide chains, as they're being produced, become inserted into the lumen of the rough endoplasmic reticulum, and then vesicles, transport vesicles that is, transfer the peptides to the Golgi apparatus, where they are further processed. they are further processed in the Golgi apparatus, and where they receive further post-translational modifications, and they are packaged for export to different places. So proteins that are destined to be secreted from cells, those are packaged into secretory vesicles, which fuse with the plasma membrane, and those proteins, which have been modified, become the vesicle opens up at the plasma membrane and releases the proteins to the external environment of the cell. Other vesicles may fuse with the plasma membrane of the cell and therefore become part of the plasma membrane of the cell. And the proteins that were synthesized originally in the endoplasmic reticulum now become incorporated into the plasma membrane. Those become proteins of the plasma membrane. As you know, there are many important proteins in the plasma membrane, giving it a variety of different properties, receptors and transport molecules, and so on. Other proteins are hydrolases, a variety of different hydrolases, which become exported as lysosomes. Lysosomes are the garbage disposals of cells. They will digest cell membranes. They will digest old, worn-out organelles. They will digest materials that are taken into the cell from the outside world that are, taken in the cell by, for example, phagocytosis. If bacteria are taken in the cell, the lysosomes containing these hydrolytic enzymes manufactured in the rough endoplasmic reticulum will be delivered to the phagosomes that are coming in from the outside. Together, the lysosomal enzymes fuse with the phagosome and digest the bacteria. In contrast, that the free lysosomes make proteins which are delivered to the cytosol or participate in formation of the cytoskeleton of the cell. So these are not membrane enclosed. They are simply delivered to the cytoplasm, okay? Now, some of them are imported from the cytoplasm into several different organelles. So some of these cytoplasmic proteins are imported into mitochondria. Some cytoplasmic proteins are imported into peroxisomes, and other cytoplasmic proteins are imported into the nucleus. But they are not first synthesized as vesicles to be delivered somewhere. They are simply synthesized in polyribosomes, delivered into the cytoplasm without being enclosed in a vesicle, and then they may do different things. For example, hemoglobin is a cytoplasmic protein which is very rich in red blood cells, and that is not enclosed in vesicles, and it's not enclosed actually within any single organelle. It's just floating around the cytoplasm. So not all the protein made by polyribosomes has to be incorporated into these several different organelles. Now, there is quality control in cells as these different proteins are made. Some of them are not good for whatever reason. They're denatured or don't fold properly, and the cell recognizes this. So proteins that are made, instead of being destroyed by lysosomes, which are more in the business of destroying whole organelles and whole organisms and larger pieces of debris, larger bits of matter, the more individual proteins are chopped up in proteasomes. So proteasomes are illustrated here. It's a barrel-shaped chemical structure, protein structure, which will break down proteins into chop up unwanted proteins into bits. So let's say there's two major ways to get rid of things that a cell doesn't want. One is to break up stuff in lysosomes, things like membranes and old, worn-out organelles. But another way to get rid of old, worn-out, denatured, misfolded, unwanted, unneeded, unnecessary proteins is to degrade it in proteasomes, like we see here. Those proteins that the cell wants to get rid of are labeled with ubiquitins, and then it's recognized by the proteasome and then removed from the cell. Okay, so here's a picture, an EM picture showing you a Golgi apparatus. They're fairly easy to recognize. They often are found close to the nucleus. That maybe is one clue, but often you can't tell it's near a nucleus looking at a particular picture, but they look like a stack of pancakes. not a nice flat stack of pancakes, one which is curling up, like if you went to IHOP and they had some old leftover. They serve, I hope they don't really. They serve you an old leftover stack of pancakes, if they want to get rid of, and they think you're not going to complain about it, and I hope you do complain about it. It may be curling up at the edges, like you see here. Well, that's the Golgi. And so materials that come from the rough endoplasmic reticulum, they're not done processing. There are transport vesicles which are delivered to the closest membrane, cisternae, flattened sac, cisternae, flattened sac of the Golgi. That closest sac of Golgi is called the cis Golgi, and then it's processed one cisternae at a time here, and then down here and down here. It's transported 1, 2, 3, 4 in a stepwise fashion until it reaches the other end. And it's finished processing at that point. Well, eventually, by the time it reaches the last stack, which is called the trans Golgi, then it's exported in a vesicle to wherever it needs to go, the cell surface, at the cell membrane, or as lysosomes, which are delivering hydrolytic enzymes, various purposes in the cell. Here's a Golgi, and this probably here, the convex surface is probably the cis Golgi, and the concave surface here is probably the trans Golgi. This is probably facing the nucleus in this area here, and this is probably facing the cell membrane in this area here. Mitochondria have a distinctive appearance. It's not hard to recognize them. If you know what to look for, they have two membranes, an outer membrane on the outside and an inner membrane on the inside. The outer membrane is nice and smooth. These organelles are often rather elongate looking. The inner membrane is folded into shelves like this. See shelf, 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 shelf often. It doesn't have to be a shelf-like fold. It could be more tubular in some mitochondria, actually, but often it has a shelf-like appearance. And there's a space between the outer membrane of a mitochondria and the inner membrane of a mitochondria, the intermembrane space. All this structure is important for what mitochondria do, which is very complicated, but put simplistically, mitochondria are the powerhouses of the cell. They are important in breaking down food substances to create energy and intermediates also, actually. But the energy is then stored in the form of ATP, which is utilized on a variety of different cell processes. It powers a variety of cell processes, ATP. So these are powerhouses that make lots of ATP. They do many other things. They're involved in apoptosis. When they release certain molecules, it facilitates the process of apoptosis or programmed cell death, and so on. So at this point in our class, we're mainly concerned with recognizing mitochondria, and there it is. Peroxisomes are another roundish-looking organelle. There are several roundish-looking organelles which can be confusing, so you'd like to know maybe how do you distinguish them? Well, peroxisomes are organelles which contain oxidative enzymes, and they, in the process of breaking things down or metabolizing things by oxidative enzymes, they often create hydrogen peroxide, which can damage cells. Hydrogen peroxide can act as an oxidizing agent or free radical, and it could damage cells unnecessarily. So these organelles often contain something which will degrade hydrogen peroxide, make it into water, like catalase. It'll break down hydrogen peroxide by catalase to make innocuous water. How do you recognize a peroxisome? Well, if you get lucky, they have a dark nucleoid crystal-like structure in the center like that. You see this more in experimental animals than you see in people, but that's a crystalloid structure. If you were to see it, it's a clue that you're dealing with a peroxisome and not another type of vesicular organelle. Another vesicular organelle, which we haven't seen yet, is the lysosome, for example. But a lysosome would not look like this, would not have that nice, neat nucleoid in the center. These are secretory granules. Again, you may think at first They resemble peroxisomes, but they don't have a nucleoid in the center. They all have something of a uniform appearance. Also, the cell type and the context and so on. All of this suggests secretory granules. It's not merely the appearance of something, it's the context, the situation you find yourself in. So, for one thing, I think they all have a rather uniform appearance. And unlike the peroxisome I just showed you, also there's lots of rough endoplasmic reticulum, which to me seems to suggest that this cell is making lots and lots of protein for secretion. So putting two and two together, here's a cell making lots of protein for secretion. After all, there's lots of rough endoplasmic reticulum, suggests to me these are secretory granules. Now, I know, for example, in pancreas, the secretory granules will be stored in the cytoplasm until they need to be released. When you don't have a meal, when you're fasting, you don't need to release digestive enzymes into your GI tract. There's no purpose in it. And there's a danger that you might release some of these digestive enzymes into your own pancreas, in which case you'll break down your pancreas and create a serious life-threatening disease called pancreatitis. So you want to keep the digestive enzymes in inactive form until they're really needed, until they're released into the small intestine and are going to be utilized in the proper environment. So what I'm saying is I see these vesicles, which look consistent with secretory granules, and then the situation furthermore suggests to me that these are the kind of secretory granules one may see in a cell like a pancreatic type of glandular cell. These are lysosomes. These are lysosomes. Now, lysosomes look kind of similar again, but you need to think about are there any features which differentiate them from what we just saw? Well, they're more heterogeneous looking. If you look carefully at them, they tend to look a bit more heterogeneous. And admittedly, it's a subtle distinction, but that's the way it is. They're a little bit more heterogeneous. But even more than that, you tend to see lots of lysosomes in a cell that's doing a lot of digesting of stuff, a lot of breaking down of stuff, a lot of catabolizing of stuff. What kind of cell breaks down a lot of stuff and catabolizes a lot of stuff and digests a lot of stuff? Well, a macrophage does. That's a so-called professional phagocyte. A macrophage, when it sees cell debris, it's a garbage collector. When it sees cell debris, it eats it up. When it sees bacteria in the environment, it eats them up. When it sees necrotic, dead material, it eats it up. So it tends to be clean up garbage and unwanted materials and pathogens. So one would expect to see lots of lysosomes in macrophages. That the context makes a big difference. Also, if you look at this particular cell here on the left-hand side, I'm not seeing lots of rough endoplasmic reticulum. And the other cell, which was a secretory cell, I saw lots of rough endoplasmic reticulum. Here I don't. So that to me suggests these are not secretory granules. Of course, you may be given other clues. This is not necessarily the only, what I just mentioned are not necessarily the only things. If a cell is breaking down its own unwanted, unneeded, worn out, unnecessary organelles, you may actually see remnants of those organelles within the lysosomes as they break that stuff down using the hydrolytic enzymes, which were originally manufactured within the rough endoplasmic reticulum.
Dr. Brown, may I
Yeah.
ask a question?
Yeah.
Can you go back a couple of slides to the peroxisome? You mentioned that if we get lucky, then we'll see the dark crystalloid structure in the middle. What if we don't get lucky and we don't see the structure in the middle? How would we differentiate?
Well, let me put it this way. I would not be able to tell what something is without some kind of help. I mean, it's too nonspecific looking. We don't create, our goal is not to create questions which are unanswerable. They should be answerable by someone of your level of training, your level of experience, and we need to use common sense. I mean, I would not put, there has to be some type of indicators in that question which a reasonable student can make a conclusion. I would not put a single gray looking vesicle in a picture and or you have to make a wild guess to get it. No.
Okay.
I
Okay.
mean,
So
I
look
have
for
to
the
say
dark,
something.
the dark
If I were to
structure.
do it, I'd say this thing has oxidative enzymes within it. What organelle is that? Or this thing generates hydrogen peroxide. There'd have to be some clue to it, right? So I'm not sure you necessarily have to worry about having to identify it simply on the way it looks. There have to be multiple pieces of information given to you. That's our job. We wouldn't, I wouldn't think it's reasonable to give somebody something and say, you have to know it no matter what. Now, if there was a nice nucleoid in the center, et cetera, like something like that, so it was so wonderful that, you could get it without being given any kind of information, okay, then it's a fair game. But otherwise, no, I wouldn't do that. I mean, I personally wouldn't put a question like that in. I don't think anybody.
I appreciate it. Thank you.
I mean, students ask, what about this potential for this kind of question or that question? The question needs to be formulated by us in a way in which you could reasonably expect it to get that answer correct. And that may mean synthesizing several pieces of information together. I say this because this is important for all our questions. It's not simply a matter of looking at a picture and guessing the right answer. It's a matter of putting everything together. It could be a piece of information in the question stem. It could be that you're given 5 answer choices and only 4 of them cannot make any sense at all, right? If 4 cannot make any sense at all, it must be the 5th answer choice, which has to be correct. So so the way the answer choices are constructed may be the clue. That's the way I would answer that question. I mean, I realize that organelle is, can be challenging to get, but I don't think you need to worry about that. Okay. Now, lysosomes, they degrade, they have lots of hydrolases. You'll see they have 40 to 50 hydrolases. If you look it up, they digest nucleic acids and they digest saccharides and they digest proteins. But the thing is, they digest as best they could. They reach a point where they cannot digest further, and the old, mostly but not entirely digested material will just sit there forever as a so-called residual body. So you sometimes see lysosomes that look like this, RB, residual body. So this is just stuff which cannot be digested further. And that's it. It's basically the end stage of a lysosome. Okay, so there are several pathways of lysosomal activity. Lysosomes, they're made from the rough endoplasmic reticulum and then the Golgi. They release vesicles containing the hydrolytic enzymes, which are now inactive. Those are primary lysosomes. Those confuse with old, worn out, unwanted organelles, which need to be, removed from the cell. That process is called autophagy. And when the lysosome fuses with the old organelle, it's that thing that's made is called an autophagosome. That's an autophagosome. Sometimes you can even recognize it. If you look at it, you may see old, pieces of cell membranes in there. Sometimes you may see an old mitochondrion, which is still being digested. Sometimes you could actually make out the fact that this is an autophagosome. But in principle, it's important, even if you don't see a picture, you should realize when a lysosome fuses with membranes or old organelle to get rid of it, that's an autophagosome. Now, sometimes, for example, a macrophage, things from the outside like bacteria or dead cell debris is phagocytized by the macrophage cleaning up garbage and getting rid of things that are unwanted. That process is called heterophagy, and that vesicle that's formed between the fusion of the vesicle coming from the outside, the phagosome coming from the outside, and the lysosome that's manufactured by the cell is called a heterophagosome. So both involve lysosomes, but they're different things in a sense. It's a different pathway. And then finally, sometimes lysosomes are made and they have within them hydrolytic enzymes to break things down. And that lysosome fuses with the cell membrane and releases those hydrolytic enzymes to the outside environment, to the interstitium, let's say, or to a body surface. So that's a little different. That's another thing which can happen where lysosomes are just releasing their enzymes to the outside, okay? So these are three simple pathways that lysosomes can become part of. Here's a picture showing you the proteasome. It's a molecular machine. It's not surrounded by a cell membrane like the other things we've been talking about. And it's a protein complex. It's too small to really see ordinarily. It's not something you see by looking at a tissue section, okay? But you know it's in there. You just know they're in the cytoplasm. You can see lysosomes, but you cannot see proteasomes. But they both are involved in breakdown of garbage that you don't want. But the lysosome is more big things like organelles and membranes and bacteria, whereas the proteasome is getting rid of damaged, unneeded, misfolded proteins. There's a set of skeleton to cells. The analogy is made to our skeleton. And there is a difference, though. The cytoskeleton is more dynamic. It's constantly changing. The filaments that make up the cytoskeleton are growing and shrinking. They're being built and being deconstructed depending on the cell's needs. They are important in not just cell structure, they help maintain cell structure like our skeleton, but they also are important in motion, cell motion. By that I mean the whole cell moving or just parts of a cell. In phagocytosis, if you saw the earlier picture, the cell was extending out like lips of cytoplasm around something to take it into the cell as a vesicle. Well, that involves movement of the cell membrane and movement of the cytoplasm to do that. The cells are extending out folds and thin processes and so on all the time as it interacts with the environment. There's all kinds of cell motions going on. Muscle cells, the whole cell contracts, the whole cell shrinks and contracts. That involves cytoskeletal elements as well. So in general, cytoskeleton involved in structure, keep maintaining the cell structure. Is it cuboidal? Is it squamous? Is it columnar? Is it something else? And movements of all variety of things. Now, we're not going to talk about the details of all those things because it's just too big a topic, but those are the generalities that you need to know. There are three main forms of cytoskeletal elements. The thinnest, smallest one are the actin filaments. Actin filaments make up the microfilament. So when we speak of actin filament, same thing as microfilament. Actin microfilaments are the same thing, okay? They are about 7 nanometers. They're the thinnest. Intermediate filaments are intermediate in their size, if you will. They consist of a variety of different proteins. It's not just one protein which makes up an intermediate filament. I said to you, microfilaments are made of actin. Yes, that's simple. But intermediate filaments can be made of cytokeratin, they could be made of vimentin, they can be made of desmin, they can be made of glial fibrillary acidic protein, neurofilament protein, lamin. Lamin is found inside the nucleus to be distinguished from laminin, which is found in basement membranes. Laminin is something found in basement membranes. Lamin is a intranuclear intermediate filament, two different things. Intermediate filaments are famous for being tough. They are the most the toughest of the cytoskeletal filamentous networks. They resist tension the greatest. They are fairly long-lived filaments. They don't break down as easily or rapidly as the other type of filaments. So they are tough with great tensile strength. And finally, there's microtubules. They're small tubules, if you will. Those are 25 nanometers. So as you see, these are defined partly on their size. One is the smallest, one is the largest, and one is intermediate in its dimension. The microtubule is made of tubulin, again, has in a way a more simple construction compared to the intermediate filaments, which can have a variety of different proteins. They have a different distribution, more or less, within the cytoplasm. The actin filaments often are found around the periphery of the cell. The outer part of the cell you may think of as being the cell cortex, if you will, the periphery of the cell. So here around the periphery, we see actin filaments, and they extend into these appendages coming off the periphery of the cell. What are these appendages? Just looking at that diagram, they look like microvilli. They're forming a core within the microvilli. In fact, this bunch of actin filaments just beneath the cell surface here are sometimes called the terminal web. So you see here the actin filaments around the entire cell periphery forming the terminal web here at the apical part of the cell. That's the apical part of the cell, and then extending into the core of the microvilli right here. So if someone does an immunostain, let's say immunofluorescence, just from the pattern of immunostaining, you can guess that those, if they look like this, those would be actin microfilaments. On the other hand, there are filaments which seem to radiate from an area near the nucleus. There's an area near the nucleus called the centrosome. The centrosome consists of two centrioles at right angles to each other, surrounded by pericentriolar matrix that's just adjacent to the nucleus in the centrosome. Again, the centrosome contains within it two centrioles at right angles to each other. The centrosome and its centriole nucleates the network of microtubules throughout the cytoplasm. So what you see here are microtubules radiating away from the centrosome. They become nucleated, but they start their life at the centrosome here, and they radiate out towards the periphery, and they try to grab onto things they want to hold on to. So if this was cell division, for example, the microtubules that are forming this network radiating through the cytoplasm will grab onto the chromosomes and then help pull the chromosomes apart during cell division. So the cell can be divided, one cell can become divided into two cells. That's one of the important functions of microtubules. Then here we have a network of cytoskeletal filaments. Here, these cytoskeletal filaments seem to link to cell junctions here at the lateral domain. Just a second while I turn my sound off here. Sorry. Here they link to these filaments are linking to cell junctions at the periphery. Just from the way these cell junctions look, scattered here and there along the lateral border of the cell, suggests to me they may be desmosomes, which are a type of adhering or anchoring junction holding two cells together. So radiating filaments linking to desmosomes in this particular case, consistent with intermediate filaments. If this was an epithelial cell, a good chance those intermediate filaments are cytokeratin filaments. If you want to know if this was a carcinoma made of epithelial cells, you could do an immunostain to see if the carcinoma contains cytokeratin within its cytoplasm, reflecting the fact that it is an epithelial tumor called a carcinoma. So these filaments often connect, as I said to you, or link to desmosomes, which are a type of adhering junction, and then they radiate throughout the cytoplasm as well, like this. They're radiating throughout the cytoplasm, surrounding the nucleus to some extent as well. So just a pattern of this dispersal throughout the cytoplasm gives us a good idea as to what kind of cytoskeletal filament is involved here. In this particular cell thing on the right-hand side, this is a mitosis. The blue are chromosomes in the center, and the green is a mitotic spindle. So I know from experience that a mitotic spindle is composed of microtubules. So my guess here is that the green is a immunofluorescent stain for tubulin making up the mitotic spindle. If you look at this, there's some arrows here. There are two poles to the mitotic spindle. There's a pole right here and a pole right here, and the chromosomes will be pulled apart towards those poles. I know that at each pole is a centrosome containing a pair of microtubules, and that centrosome with its pair of microtubules nucleates the microtubules as they radiate outward from the centrosome. So apparently, the original centrosome of the cell replicated into two and then migrated to the two poles of the cell like this to form a mitotic spindle to pull apart the chromosomes. Here we seem to have a network of filaments staying red with immunofluorescence, and there's a more densely stained area here right next to the nucleus. This could very well be the centrosome containing those two microtubules again in pericentricular matrix, and they're radiating out into the cytoplasm like this. So these are probably microtubules as well. It's a different stain, but radiating throughout the cytoplasm. This on the left-hand side is not a mitotic cell. So of course, you're not going to see a mitotic spindle in this case. Here we see another cell that's been immunostained with immunofluorescent staining. There's a nucleus. The nucleus has a dye, which makes it look blue here. And you see two different colors. On one color, there's a circumferential rim of kind of orange-red staining filaments because of the appearance seems to be staining the cortex of the cell, right? The outer part of the cell. This is highly suggestive that these filaments staying on the outside here, the circumferential ring on the outside, are actin microfilaments. Actin microfilaments, okay, just from the appearance. And according to the information given us, the green consists of microtubules. I probably would have found it a little bit challenging, but this is from the textbook and they label it clearly as such. So I'm going to accept that these are microtubules. They seem to start near the nucleus, probably in the centrosome, and they seem to radiate outwards like this from the nucleus towards the periphery. So that's perfectly consistent with microtubules being nucleated in the centrosome, radiating out to the periphery. And then the circumferential ring like this is then consistent with the actin microfilaments.
Dr. Brown?
Yes.
I have a quick question for you about the intermediate filaments versus the actin filaments. How would you, is there like kind of a hard and fast way you can really tell the difference? Because I could see there being a little bit of confusion with the intermediate filaments maybe coming from the side of the cell and kind of branching up and around and maybe looking a little bit like the actin filaments.
Well, in this particular picture here on the screen, this is both of those together, intermediate filament. Excuse me, no, these are microtubule. Well, the difference, let me put it's similar to this, okay? The solution is similar to what we're seeing here. The actin filaments form this circumferential band around the periphery. The intermediate filaments would be radiating through the cytoplasm. They probably would be radiating through the cytoplasm in a less discrete way.
And they're not going to be nucleolar they're not going to be associated with the nucleus like microtubules would, though, right?
Well, microtubules tend to nucleate at the centrosome and radiate outward. Of course, you have to have a good section and it has to be well oriented, all that kind of stuff. But
right.
I wouldn't worry about it for your
yeah.
for your purposes. I would not worry about it because
okay,
we would, ordinarily we would not put in something which would be excessively challenging. When something is challenging, it's challenging for us too. It's not because we can't tell, but teaching samples are based on fairly idealized pictures, things where things work out in a very standard, recognizable kind of way. You wouldn't want to put something that deviates too much from a standard, fair, easily recognizable kind of preparation. So I wouldn't worry
Thank you so much.
about it. But I mean, I know, for example, there are EM pictures where maybe in the epithelial part of the course, where you see filaments linking to the desmosome, okay? If I see filaments linking to a desmosome on EM, I would say intermediate filament. But you don't have that issue in this particular picture. This picture has nothing to do with that. Here, I clearly see a circumferential band of filaments right beneath the cell membrane. It makes total sense that this is actin.
That helps. Thank you.
Okay. I mean, a lot when things are not ideal, of course, they are challenging, but they're challenging for anybody. But our goal is not to give you a unusually challenging things. Our goal is to give you things which you could reasonably be expected to get. Now, I'm not saying we necessarily will always be successful in that endeavor, but that's our goal, of course. Yeah. Okay, let's talk about the nucleus. This is tissue section of brain, and we see nuclei which are rather pale, like we see here. They're pale because they have lots of euchromatin, chromatin not very condensed. Here we see nuclei which are dark, like here. These nuclei have lots of heterochromatin, chromatin which is a lot more condensed. So in general, when you look at nuclei, they could have lots of euchromatin or lots of heterochromatin. Of course, nuclei always have some of both. It's not like, or almost always, let's say, they almost always have some of both, but often one or the other will be predominating. Here's EM showing you a nucleus, and you see lots of pallor. That's where euchromatin, uncondensed chromatin, is located. And then just beneath the nuclear membrane is heterochromatin. It's more condensed and therefore more electron dense and therefore more dark. And here's a big mass like that. That's the nucleolus. So the nucleolus stands out from the other things that you see in the nucleus by being a discrete structure that has some distinctive features, okay? So heterochromatin here, heterochromatin here, heterochromatin there, heterochromatin there, nucleolus here. Now, to be sure, there is some heterochromatin also binding to the outside of the nucleolus as well. This is heterochromatin right there. Okay. Now, it so happens when you have two X chromosomes, like a typical phenotypic female, one X chromosome will condense almost entirely, and the other X chromosome will be, normal, like other chromosomes, let's say, have large areas which are not condensed. So that condensed chromosome in someone who has an XX pattern of chromosomes is called a bar body, That occurs in a typical phenotypic female. So here's a buccal epithelium. If you scrape the cheek cells, the buccal epithelial cells off and stain them, do a cytologic preparation, stain them, you will see a little clump of chromatin, condensed chromatin that is. That's condensed chromatin. Attached to the nuclear membrane, it stands out because it's bigger and more condensed than most of the remaining chromatin in the nucleus. That's a bar body. There's a bar body. There's a bar body. That's a nucleolus. That's a nucleolus. That's a nucleolus. This is chromatin, which is heterochromatin, okay? But it's not a bar body. A bar body is that somewhat discrete mass. That's a single condensed X chromosome. That's a single, there it is, a single condensed X chromosome. Now, if you took a sample of blood, did a blood smear, staining with the usual blood stains, which are not H&E, they're different stains we haven't talked about. A blood smear type stain. You'll see white blood cells like this. This is a white blood cell. It has a lobulated looking nucleus, and there's a drumstick seemingly attached to the surface of the nucleus. See that? It looks like a drumstick. Think of a drumstick. That's the bar body in a white blood cell, a neutrophil or polymorphonuclear leukocyte. Same thing. Neutrophil or polymorphonuclear leukocyte has a bar body often, which looks like a drumstick. So these are both bar bodies. They look a little bit different in different cell types. You see this in an individual with two X chromosomes. So of course you can see it in females, and you don't see it in XY males. The Y chromosome makes a male individual, male. It gives them the male sex characteristics. Now there are karyotypic abnormalities. For example, someone could have, they could be a phenotypic male. They have a Y chromosome, but they also have an extra X chromosome. There are a 47XXY male, 47XXY male, because during meiosis, the chromosomes didn't separate normally. Normally, the X and Y chromosomes separate away from each other, but they don't separate away from each other properly. If there's non-disjunction, a person may end up with two X chromosomes and one Y chromosome. So they're phenotypic male, but they are somewhat more feminine, let's say, than a typical male. They have small testicles. The volume of testicles may be abnormally small. They may have gynecomastia. They may have some other characteristics like they're unusually tall, for example, things like that. That's called Klinefelter syndrome, a 47 chromosome XXY male, and they will have a bar body. That person with Klinefelter syndrome, XXY, will have a bar body on cytologic preparations. If you did a karyotype, you will see two X chromosomes and one Y chromosome. So you can't tell right now looking at these cytologic preparations, but on a karyotype, two X chromosomes and one Y chromosome on a karyotype of somebody with Klinefelter syndrome, small testicles, gynecomastia, some other features as well. Cells can be grouped by regenerative capacity. Some cells divide all the time, like cells in, let's say, a dangerous environment, like the intestine, divide they have stem cells which are dividing all the time for the entire lifetimes. They need to constantly reconstitute the cell population. The intestine is full of all kinds of potentially dangerous and noxious stuff like bacteria and viruses and environmental substances, which can cause problems. Other cells divide all the time, like bone marrow. You need to constantly replace your blood cells all the time. So there are hematopoietic stem cells in bone marrow that are labile. Many of the viscera and other cells of our body are stable. They they don't need to divide all the time, like liver, most cells of kidney, the cells which line blood vessels, and so on. Those cells can remain stable. They could just exist for, quite a long time without necessarily dividing and replacing themselves. Although if there's a need, they could go into the cell cycle and divide. So labile cells are always in the cell cycle. They're going from G1 to S to G2 to M to G1 to S to G2 to M. Stable cells remain, they exit, they take a rest from the cell cycle. They become quiescent for the most part. So they go into what's called G0. They're quiescent. So a liver cell doesn't need to divide all the time, goes into G0, it just sits there as long as it possibly can. But then if it's needed, it could go from G0 into G1 and start dividing again in the cell cycle. Now, there are other cells which are permanent cells, things like the neurons of the brain and cardiac muscle cells of the heart, which are end-stage cells. They remain in GO. They are outside. They're not cycling all the time, in a sense. They're not in the cell cycle, in a sense. They are in G0 phase. They just remain out, basically non-dividing for a person's entire lifetime. And the problem with that is if you lose neurons, if you lose cardiac muscle cells, you can't replace them easily. So, but it's important to realize then that the cells can be in these three different populations. So the cells which are able to divide, and they will be in the cell cycle. The labile cells always in the cell cycle. The stable cells, they remain quiescent in the GO phase for quite a while, but then they could go back into the cell cycle, divide. Ultimately, neurons, cardiac muscle cells, always in G0. They will not divide. That's it. You're stuck. Unless we find a way around that limitation, of course. It's good to know the phases of mitosis. PMET, prophase, metaphase, anaphase, telophase. Easy to remember. It's good to be able to recognize these different mitotic phases. When the nuclear membrane breaks down and the chromatin or chromosomes condense, so they're now visible. Normally they're not visible. They're too thin. They're very thin. Chromatin is very thin normally. When the chromosomes really condense to become very thick to the maximal extent in mitosis or meiosis, they're visible. So here's prophase, where the nuclear membrane is broken down in preparation for the cell to divide into two cells. You see these thread-like chromosomes in a ball, forming a ball. So this is prophase. Here's metaphase. Those chromosomes in the ball then get grabbed by the microtubules of the of the mitotic spindle like this. You see here, a mitotic spindle is formed. The nuclear membrane is broken down. The centrosome is replicated into two centrosomes and separated from each other. The two centrosomes have separated from each other. Inside the centrosome here are two centrioles at right angles to each other. They're nucleating the microtubules of the mitotic spindle, and they're binding via kinetochores actually to the chromosomes. This phase is called metaphase, at the equator of the cell. That's the chromosomes are all at the equator of the cell. And here we see the chromosomes are beginning to separate away from each other.
I need that
Separate away
to
from each other.
end so that they can send the recording. Yeah. Well, my thing is. My thing is,
Okay, so this is anaphase where they're separating away from each other. You still see the mitotic spindle and you see the poles of the mitotic spindle. Okay. And here they've gotten pretty much to the end of their separation. They're now at the poles of the mitotic spindle, and the cell is beginning to separate into two, the cytoplasm that is. To two, you see this pinching off of the cytoplasm periphery like this. They're separating the two cells. So this is telophase. Here we see immunofluorescence of the mitotic spindle. We saw this earlier. These are microtubules. The protein for the spindle here, the green part of the spindle is presumably tubulin. And here we see EM of the mitotic spindle. In the center, we have the chromosomes, which are condensed at the equator. So this, I presume, is metaphase. And here we see the centrosomes containing two centrioles at right angles to each other, which form the poles of the spindle on electron microscopy. The threads here are the microtubules of the mitotic spindle. This just shows you the mitosis, and there's meiosis. We're not going to talk too much about them, but meiosis consists of two phases separated by in which there is no replication of the chromosomes between the two phases of meiosis, meiosis 1 and meiosis 2. So the end result of mitosis
I have a quick question.
two cells with the full chromosome content. The end result of meiosis are 4 haploid cells. Okay. Yeah.
Dr. Braun?
Okay, go ahead. That's the last slide. So go ahead.
Oh, so sorry. So I had a quick question regarding the bar body. So I know when we were learning about it, we learned how the bar body is considered and I'm so sorry, I'm probably pronouncing this wrong, facultative cell. And that means that it coils and uncoils as well. But how does that work? Because I thought that the bar body had to be like inactive at all times. So why would it fall under that category versus constitutive?
Yeah, that's confusing, isn't it? Well,
Yeah.
some in a woman with 2 X chromosomes, some cells have condensed, that almost totally condensed chromatin. Some cells have ordinary chromosome, an X chromosome. The other X, which is so one X chromosome in a cell can become condensed, and the other X chromosome can remain open. So the fact that one X chromosome condenses and the other in one cell and in the other cell remains open means that that chromatin is not always condensed. When chromatin is always condensed in every single cell, that's constitutive heterochromatin. What is constitutive heterochromatin? Well, the centromere is constitutive heterochromatin. The telomere is constitutive heterochromatin. That chromatin is always condensed in every single cell. It doesn't express itself by making RNA and protein. That chromatin is structural. It's always condensed. In contrast, the X chromosome of a person with 2 X chromosomes can decide, I'm going to remain open or I'm going to condense in any given cell. Because it's capable of remaining open or condensing in any given cell, that's a facultative heterochromatin.
Perfect. Thank you so much. Appreciate it.
Yeah. Anybody else?
I had a question regarding like just like on a slide, the difference between the rough ER and a mitochondria. They look very similar to me. So how would you differentiate that?
Well, I think the best way to differentiate them is to look at them a few times side to side and compare them. That's the best way, because you could think about all you want, but here's rough ER, okay? It's a it's a cisternia, a thin flattened sac. Imagine a sac, flatten it out. And on the surface, you have granules, which is the ribosomes, right? Okay. Now, let's see if we can find a mitochondria. Here's a mitochondria. This is one single ovoid organelle with shelf-like cristae going across from one side to the other. This is a discrete organelle. These are not stacks and stacks of flattened sacs. This is a discrete ovoid organelle containing shelves. That's the way I think of it. Now,
That helps a lot. Thank you.
that description may be okay, but it doesn't work better than just looking at them side by, more than once. Look at it side by side, compare and contrast, do it more than once, do it a few times. It'll start to sink in after a while. Anything else?
Thing.
Yeah.
Could you briefly explain to me again what polyribosomes do?
Let's see if I get here. Well, a thread of mRNA has attached to it multiple ribosomes. Because of that, it's called polyribosome or free ribosomes. This is the apparatus for making protein. That's it. They make protein. Now, when they remain free in the cytoplasm, they make cytoplasmic proteins. That's it. They make cytoplasmic proteins. Now, in some cells, like a red blood cell, it makes hemoglobin. Hemoglobin simply stays in the cytoplasm as a protein in the cytoplasm. But some of those proteins that are made to be released in cytoplasm have a signal in the peptide to become imported into mitochondria, peroxisomes, or into the nucleus. So some of those have a signal sequence, which will lead them to be imported into mitochondria, peroxisomes, and nucleus. But that protein was never inserted into a vesicle. It was never inserted into a vesicle. In contrast, with rough endoplasmic reticulum, these proteins are inserted into the lumen of rough endoplasmic reticulum, and transport vesicles butt off of the rough endoplasmic reticulum, migrate to the Golgi apparatus, where they're further processed and for exporting somewhere. They're further processed, and some of them will be exported to the cell membrane. Some will be released, secreted into the interstitium around the cell. Some of those proteins will be in vesicles will become basically what it's called a primary lysosome. They can